Hi everyone, my name is Francesca Susca, and for whom does not me yet, I'm a global pet development manager within Lallemand Animal Nutrition. I'm extremely glad to have been invited once again to such a great event, and I hope next time will be the right one to meet all of you in presence. Today, I will speak about the impact of the urbanization trend on pet health and well-being. More in details, we will see how this trend will affect the oxidative status of our beloved dogs and cats. But let's start from the global mega trends. We are all familiar with the climate change, resource scarcity. So there are several trends that are literally reshaping the world we live in and that are somehow connected one each other. We're living in a world that is more populous, more prosperous, and there is a higher demand of water, food, energy. So of course, we are facing resource scarcity. Together with that, we are also living longer and we need to be also happier and healthier. So another big mega trend is healthy living. We are all seeking to live healthier. Amongst those global megatrends, no doubt there is urbanization. And you can see here in this picture, in this graph, you see the two lines of urban and rural portion of people living in the world. And in 2009, the world's urban population has surpassed the proportion of people living in urban areas. And you can see the green line is keep rising, rising, and rising. The global mega trends of urbanization, together with some other more specific pet trends, such as pet humanization, changing of pet owner demographics, are inspiring the innovation that we are requested to bring on the market. But you're probably wondering how such a trend like the global urbanization may affect the pet health and well-being. Well, that's not just you wondering about that, but it's already a few years that scientists are interested in the impact of urban environment on pets' health and well-being. And it has been recently recognized that the urban areas may be hectic and stressful environments to live, as they are full of stimuli predisposing dogs to fear-related and other behavioral problems. In 2020, in such a nice paper I'm sharing here, scientists have found a novel association between the living environment of dogs and social fearfulness. And it's well represented in the graphs here on the right. They have seen that dogs living in a more urban environment were more likely to be afraid of other dogs or strangers. So have you never thought on the impact of high level of stress on pets? Well, I can tell you that there are some effects of stress that are quite visible, such as behavioral problems, but there are some others that are less visible, a bit hidden, but nevertheless highly dangerous. We all know that the exposure to stressful situation brings to biochemical and molecular changes that can emerge at cellular level and lead to oxidative stress. I like to show the picture here on the box, in which you can see how looks a normal cell and how the same cell looks under free radical attacks, and how on the extreme right the cells looks under oxidative stress. So you can easily understand that the cell under oxidative stress is going to lose its functionality, and that's not what we want from our cells and our organs and our body. And to visualize it even better, these oxidative stress on a day-to-day life situation, let's look at this apple. I'm sure that most of you have seen once in their life an apple on the table going towards oxidative stress. That's how it looks like, and that's how organs under oxidative stress would look like. So what I want to say is that we need to avoid stressing situation that could lead to oxidative stress, but we know that in the day-to-day life, we're gonna face stressing situation. So let's try to help ourselves, but of course, in this case, we are talking about pets. So let's try to help pets in maintaining the oxidative homeostasis, so in being able to maintain a good balance between pro-oxidant and antioxidant molecule that would counteract oxidative stress coming from stressful situation. Well, how to do that? That's a good question. And I would say that to reply, we need to start from the really beginning. We need to know which are reactive oxygen species, what are free radicals, what are antioxidants, why I should use them, and which one to use. So let's go step by step. The reactive oxygen species, they are reactive chemical species containing oxygen. In a biological context, they are formed as a natural byproduct of the normal metabolism of oxygen, and they have a quite important role within the cells in cell signaling and homeostasis. However, during times of environmental stress, the reactive oxygen species level can increase dramatically, and it results in significant damage at cell structure. Within the reactive oxygen species, there are free radicals. You can see them on the right side. And free radicals are atoms or molecules having one or more unpaired electrons in the most external layer of electron. And these unpaired electrons make them unstable. So they can damage the cells at various levels, and in excess, they lead to oxidative stress. So how can I do to fight against reactive oxygen species and free radicals? Well, I have to use antioxidants. Antioxidants are those substances that are able either to metabolize reactive oxygen species, transforming them into harmless molecules such as water and oxygen, or those substances that are able to stabilize free radicals by donating them an electron. So antioxidants, they are able to protect membrane and cytosolic components against free radical damage. So they are really the major defense against oxidative stress. As I just mentioned, reactive oxygen species, they are produced physiologically in the mitochondria. The mitochondria are those cell batteries that provide energy. They control apoptosis, so they are quite important within a cell. And they are the major physiological source of reactive oxygen species. It has been estimated that 90 to 95% of the oxygen passing through mitochondria is converted into water, while the remaining 5 to 10% is reduced, which creates reactive oxygen species. Within the cell, the reactive oxygen species generating system is coupled to an antioxidant defense mechanism, which represents the most important endogenous defense mechanism against oxidative stress. Of course, if the antioxidant system is not sufficient, then more ROS, more reactive oxygen species, are produced, and we're going to have damages. So we have just seen that the reactive oxygen species are produced physiologically in the mitochondria by the metabolism of oxygen. But there are some other not physiological events which can cause the production of reactive oxygen species. Those factors are environmental factors such as pollution, radiation, but also the urban environment, as we have seen, is full of stressful stimuli for the animal. Can also be poor diet, age, or certain conditions such as inflammation, because we know that inflammatory cells are able to produce high level of reactive oxygen species. But we have just seen that the ROS production system is coupled to an antioxidant defense system, which is really helping in counteracting the production of ROS and is reducing the oxidation of DNA, the peroxidation of lipids, so all the damage coming from the reactive oxygen species production. And it happens when there is a good balance, the right balance between pro-oxidant and antioxidant. And the right balance is a balance in favor of antioxidant molecules, because a balance in favor of antioxidant molecules, it's gonna support the maintenance of oxidative homeostasis, which is what we want. In some other situation, there is an imbalance between oxidant and antioxidant molecules, which is in favor of pro-oxidant, maybe because the antioxidant system defense was not strong enough, or because there was such an excess of ROS production that the antioxidant defense was not able to counteract. And in this case, we're going to have oxidative stress. Which are the consequences of oxidative stress then? Well, the consequences of oxidative stress are at a different level. We know that oxidative stress is detrimental at molecular level because it brings molecular damages like degradation of fatty acid, lipids, DNA, and those molecular damages, they reflect into cellular damages because we're going to have alteration of cellular membrane. The cells will lose their functionality. They can go towards apoptosis and so on. These molecular and cellular damages, they reflect into more physiological damages. So a dog under oxidative stress would looks in a poor healthy status. A cat would have deficiency in the immune system. The oxidative stress can have impact on fertility and the reproductive performance and so on. We can have puppies and kittens not growing very well or with a low food intake and so on. And then we have damages that are more systemic and long-term, really disease such as cancer or dermatological problems, allergies. And the scientists in the last decades, they are really focusing and working and supporting, showing that there is a link between free radicals production, oxidative stress, and disease at various levels in different organs of the body. Of course, the research is much vast and wide in humans and lab animals, but in the last few years, there has been an increase of research on the link between oxidative stress and specific disease in dogs and cats. I have chosen a few of them to show to you without going too much in details, but just to highlight how dangerous oxidative stress can be and how important is the maintenance of the oxidative homeostasis. Let's start with cats because we always speak about dogs, and also myself, I always speak about dogs. So I want to start really from cats because they are important, and we know also the population of cats is increasing, and most of us has at least one cat at home. In human, oxidative stress is recognized to be involved in the progression of chronic renal failure. So scientists have speculated that cats under chronic renal failure have increased oxidative stress. So in these studies, they looked into some oxidative stress parameters in 20 cats that were diagnosed under chronic renal failure previously, and in 10 cats of the control group with the same age. Let's look at a couple of parameters like the ratio reduced to oxidized glutathione that was significantly greater in the cats under chronic renal failure. What does it tell us? It tells us that in cats under chronic renal failure, there was an activation of the antioxidant defense system that was trying to counteract to oxidative stress, to the increased production of reactive oxygen species. And you see also here the antioxidant capacity. You see that the antioxidant capacity of cats, sick cats, those cats under chronic renal failure, was much lower than the control cat. Of course, because those cats were really fighting against oxidative stress. They were using all their antioxidant defense to try to counteract the ROS production. So in conclusion, just looking at those two parameters, what we can say that a proper antioxidant supplementation strategy in cat under chronic renal failure is a potential therapeutical intervention to restore the oxidative homeostasis, and it's really key. Another study I want to bring here is about obesity in cats. Obesity is increasing in human, is increasing in pets, and the reason are mainly two. A high ingestion of palatable and nutritious food from one side, a sedentary life on the other side. And probably the urbanization trend is even increasing these this problem because pets, they live in small apartments. The pet owner, when they come home from work, they don't really feel to spend hours in exercising, so they are really much more sedentary, and it brings to accumulation of adipose tissues, and the result is that more than 25% of the cats in the world are obese. So in the study I'm going to show here, scientists, they looked into some oxidative stress biomarkers and inflammatory markers of healthy cats during their optimal weight, during the weight gain, and during the maintenance of obesity. Here you see they look into the markers of inflammation like interleukin-6, which is an acute phase inflammatory cytokine, and in the C-reactive protein, another inflammation biomarker. For you to better understand, time 0, 1, 2 is time 0 is the optimal weight. From time 0 to time 1, there is the gain weight of the cat. And from time 1 to time 2 is really the maintenance of this obesity status. And you see that both the CRP and the interleukin-6, they are dramatically increasing from time 0 to time 2, so throughout the weight gain and the obesity maintenance, which is suggesting a situation of chronic inflammatory response to the weight gain and obesity. As a marker of oxidative stress, they looked into the malonaldehyde level, which is really a marker of lipid peroxidation. And so they have looked at it at time 0, so optimal weight. They've seen almost 3 times higher during the weight gain, and still quite high also in the maintenance of obesity. It means that during the weight gain and obesity, there is really a damage to the tissue due to the oxidative stress, so to the inflammatory cells that were producing higher level of reactive oxygen species. So again, antioxidant supplementation would help here to maintain a good oxidative status and to reduce the damage caused by oxidative stress. But obesity is not an healthy concern just of cats, but as we said, is also of human and dogs. So there is for the same reason, because they are consuming a lot of food, they are much more sedentary. And in this study, what they have looked is, I will make it quite easy. So they look before and after the supplementation with the food that was enriched in a natural antioxidant. And they look in several parameters, but the first 3 are quite interesting because was the body condition score, which of course was decreased in after these food enriched with the antioxidant, but they look also in derivatives reactive oxygen. Again, they were significantly decreased in dogs after the supplementation with these food rich in antioxidant. And even more interesting, you see the biological antioxidant potential of those animals was much higher after the supplementation with the antioxidant. So really, it tells us that a proper diet with a special emphasis on the antioxidant addition, it's crucial from one side to maintain the ideal weight, which is something that we really want, and also to support a good oxidative balance, so the good oxidative homeostasis that we are looking for. Cognitive function is the last study I'm going to show you, quite interesting because antioxidants also in humans are used as anti-aging, both topic on the skin or systemic with systemic supplementation. Why they are important? You see here in this study, in this picture, you have in the early adult life, there is already around 30 years old quite important decrease in SOD activity. SOD is a superoxide dismutase, so it's a primary enzyme. We will see it later, so I just not going details now, but I want you to bear in mind the superoxide dismutase as an endogenous primary enzyme, quite important, and its activity is seen to be decreasing already in the early adult period. That's a study on human, but I would expect exactly the same to happen in dogs. And brains of aged dogs, they accumulate oxidative damage to proteins and lipids, which may lead to dysfunction of neuronal cells. The production of free radicals from one side and the lack of increase in compensatory antioxidant enzyme from the other side may lead to detrimental modification to important macromolecules within neurons. So, based on previous research, but also on results by lab and clinical studies, antioxidant may be a class of nutraceutical that, reducing the oxidative damage, are able to significantly improve or at least slow down the decline of learning memory in dogs. That's what this interesting study was telling us. And so how now what we have seen that in these in several studies in cats, in dogs, how really the oxidative stress and the damage caused by an increase of oxidative stress can impact pet health and well-being. Now we have to see how we can work on that with antioxidant and which could be the best strategy to help pets in maintaining oxidative homeostasis. Once again, we, as I like to start really from the beginning to try to really follow a flow and understand exactly what we need to understand to then find the right strategy. Let's start again also here from the really beginning, which is the superoxide. The superoxide is the one that you see in the red circle, is the major free radical that is produced during physiological and pathophysiological state. It is scavenged by the superoxide dismutase. I told you right a few seconds ago, bear in mind SOD. SOD, as I said, is a primary antioxidant, is an endogenous enzyme, is a superoxide dismutase, is the only enzyme able to transform the superoxide into hydrogen peroxide. The hydrogen peroxide is, it's not a free radical anymore, but is a reactive oxygen species. So still harmful, still need to be transformed into harmless molecule like water and oxygen. It happens thanks to other two primary antioxidants, which are the catalase and glutathione peroxidase. Those three enzymes, they represent the primary antioxidant chain. So they are able to decrease the free radical production upstream. They have kind of preventive effect. So they really take the superoxide, the most harmful free radical, and bring it into harmless molecules. Of course, that happens. What happens is that few of them, either because there is a lack or deficiency of SOD or primary antioxidant, or there is an excessive production of superoxide, what happens is that you still have production of some free radicals. And then whenever you have production of free radicals, what you can do is to use secondary antioxidant. You need the secondary antioxidant like vitamin E, C, A, and those secondary antioxidants, they work downstream, so they are able to neutralize free radicals once they've been already produced. So you have a primary chain that works upstream, you have a secondary chain that works downstream. What would be the best is to use both of them in order to target the different step of the oxidative pathway. So you're going to have, with the first chain, you're going to reduce the production of free radicals. With the second chain, you're going to neutralize the few free radicals that have escaped the first line of defense. This is the pyramid, the antioxidant pyramid. It's quite interesting and quite easy to understand. It helps us really in understanding the different antioxidants that are available, that I can use, that I can blend, that I can put together really to counteract oxidative stress. Let's see, at the base of this pyramid, we have the cofactors, yes, the trace elements and proteins, which are cofactors of primary antioxidants. It means they are not antioxidant by their own, but they are needed by primary antioxidant to work. On top of them, we have the secondary antioxidant. We have just seen them now. They work downstream, they are able to neutralize free radicals, and they are in a ratio 1-to-1 with free radicals. It means one secondary antioxidant is able to neutralize one free radical. So they are quickly saturated, I would say. On the really top of the pyramid, we have the primary antioxidant. So those endogenous enzymes, which are the superoxide dismutase, the catalase, the glutathione peroxidase, which are really working upstream by preventing the formation of free radicals. As I said, they are produced endogenously, and they stay in a ratio 1-to-1,000,000 with the free radicals. It means that one primary antioxidant is able to metabolize more than 1,000,000 of free radicals. We don't have a lot of time, but I just want to go through each of them of these big categories, so cofactors, secondary and primary antioxidant, to give you a few additional information that I believe are quite useful. Secondary antioxidant, so right in the middle of between primary and cofactors. Secondary antioxidants are the most commonly used in food, in pet food. We see those are the main one that are used to support antioxidant claims. They are quite useful. They come from the feed. We have, I choose a few of them. They are quite common. We are all familiar with them, like the vitamin E, which is a chain-breaking scavenger. We have polyphenols, which is a huge family of more than 8,000 different organic molecules, and they can catch free radicals produced during oxidative stress. Within polyphenols, we then have the subclasses of flavonoids, which they are able to scavenge free radicals by donating them an electron. So we have seen they work downstream in a 1-to-1 ratio. Trace minerals, they are, as in the title, quite efficient micronutrients that have several metabolic immune functions. And a few of them are really important to minimize the effect of excessive free radicals production. Of course, I'm going to focus on them since we are talking about oxidative stress and how to counteract it. And a few of them are quite important cofactors of antioxidants, such as copper, zinc, manganese, or selenium for the glutathione peroxidase. And it's exactly about selenium that I would like to spend a few words. The selenium is quite an important trace mineral. It's part of more than 25 functional selenoproteins, and so it has a lot of metabolic function in the body. Today we speak about the oxidative stress and we speak about primary antioxidant, and well, selenium is part of the glutathione peroxidase, which is a selenium containing group of antioxidant enzyme made of 8 enzymes. Out of these 8 enzymes, 5 of them are selenoproteins, so they contain selenium. So we have seen it. They are really involved in the detoxification of the reactive oxygen species hydrogen peroxide, and will help it in transform into harmless water and oxygen, so harmless molecules. So quite important in the oxidative stress situation. Last things I want to say about selenium is should I use it organic or inorganic? That's something sometimes it's a question that I got from customers. It's important to know why I should use organic or inorganic. Well, if we think about wild dogs and cats, of course, what are they using? They were used to eat animal tissues, so they were used to eat organic selenium that was coming from this animal tissue. And also the main feed materials such as grains, oil, oilseed, they all contains organic selenium, and more than 50% of it is made of selenium methionine. So it's known, is recognized that the organic form of selenium is the most bioavailable form. And the intestinal tract is optimized for the usage of organic selenium, which appear to be even much lower toxic, even if used in a high level. But still, what I see on the market is the main source of selenium use is the inorganic form, like sodium selenite or sodium selenite. But probably not all are aware that the inorganic form of selenium could be harmful. And you can see here in this picture on the left side, you see the intestine, a gut of a control chicken that shows normal duodenal cells in the gut wall with the crypts and the glands. While on the right side, you see a picture of a gut, a duodenum of a chicken that has been treated with high level of inorganic selenium. And you can see here a vacuolar and hydropic degeneration of the epithelial cells. So the suggestion, of course, is to use organic form that are also quite available on the market, even in the form of enriched yeast. So yeast that has been grown on a substrate of inorganic selenium, and while growing, the yeast was able to incorporate the selenium and metabolic transform it into the organic form. But let's come to the top of the pyramid. On the top of the pyramid, we have the primary antioxidants, so superoxide dismutase, catalase, glutathione peroxidase, which are essential enzymes. They have the highest protective effect. They work upstream by detoxifying free radicals. So the superoxide dismutase is the only enzyme able to transform the most powerful free radical, which is the superoxide, into water and oxygen. So of course, before in hydrogen peroxide, and then in water and oxygen through the other two primary antioxidants. So the superoxide dismutase, it's really a key enzyme because it's really the first one that is starting the reaction of detoxifying the free radical. Where I can find it in nature? Well, SOD can be found in nature in several ingredients: algae, pea seed and leaves, in spinach, in rice, in maize, in tomato leaves. But there is a very high concentration of SOD in a specific variety of melon. And the way it has been discovered, it's quite interesting, and it's already saying a lot about its potential in several applications. It was the 1989 when a specific variety of non-GMO cantaloupe melon have been discovered to have a much longer shelf life if compared with the traditional variety. You can see it here on the top, the traditional variety, and here on the bottom, the specific variety of cantaloupe melon. And in 10 years, there has been 10 years spent in optimizing this variety to meet agricultural yield and production process. But today is the natural vegetable source with the highest content in SOD and with an enzyme activity which is 20 times higher than the traditional variety. The original mechanism of action of Melafid is based on the stimulation of the endogenous production of the three antioxidant enzymes that now you are very familiar with: the superoxide dismutase, the catalase, the glutathione peroxidase. And the mechanism has been demonstrated in several organs, such as muscles, heart tissue, liver, adipose tissues, and also the reproductive tract, and in several animals, like rodents, laying hens, swine, chickens, and so on. So it's really implying that there is a common process shared by all the aerobic organisms. I show here just a few of those study and demonstration. And here on the left, you have a demonstration of increased production of primary enzyme in the heart of mouse that was really reducing cardiac hypertrophy. While here in obese hamsters, there was the melon sod supplementation was able to reduce adipose tissue. And so really there are several studies that are supporting the evidence that the supplementation with melon sod was increasing the endogenous production of primary enzymes. But how does it work? First of all, it's important to know that, as we said, the superoxide dismutase is an enzyme, which means it's a protein, so it needs a coating. A coating is essential to prevent the sod to be denaturate and so lose their effect due to the gastric juice passing through the stomach. So we want it to bypass the stomach, and once the coated melon is able to reach the ileum, in the ileum it interact with some receptors called lectin receptors, which are able to induce, to stimulate an increase of endogenous production of the primary enzymes. So it's important to understand that there is not really an absorption of the SOD, but the SOD is able to stimulate receptors that are inducing an increased production of primary enzymes all over the body. And these enzymes, these SOD, glutathione peroxidase, and catalase are really the first line of defense against free radicals, and they are able to inhibit oxidative stress and its pathway. Well, we, of course, as I showed, there are several studies on different animals, but we really wanted to show and to support the positive effect of such a melon sod on pets. But we know that we really don't want to have negative impact on pet well-being, so we needed to find a stressful situation which was not really impacting their well-being. So what we thought is to try to see the effect of melon sod in helping sport dogs, like racing dogs, in better managing physical and mental stress. So we did work in collaboration with the University of Paris, where we with them we looked into the melon sod and their effect in reinforcing the total antioxidant status of racing dogs during and after intense physical activity. So we used in this study 18 Alaskan Huskies that were supplemented with the melon sod 13 days before, a couple of weeks before the race. We did a blood sampling at T1, so the day before the first race. Then the dogs had 3 races in a row, so 3 days of racing. And the day after, we got another blood sampling just to check the total antioxidant status of those dogs right after the very high intense physical activity. And we got another sample 1 week after. So let's look at the result. We see here in the results that you see the green bars are the dogs that received the melon sod supplementation. And as you can see, after the 3 days in a row of racing, the dogs supplemented with melon sod, they were showing a significant better total antioxidant status, which is really showing the fact that the dog were able to manage a stressful situation, which was a competition is always a physical and mental stress, much better than the control group. In the week after, the recovery was good for both the group, and was expected, because all the dogs we work with were very well exercised dog, were used to do very hard training. But it's good to see that also with this hard trained dog, the stress was managed much better by the dog under melon sod supplementation. So we were really able to support that melon sod supplementation reinforce the antioxidant defense of pets in challenging conditions. Good, so I'm gonna conclude. I want to remind you that free radicals production is a physiological process, so it happens, but normally in normal situation it's well regulated by the endogenous antioxidant defense. But of course, free radicals production, there are situations which can really exceed, so we need to help our pets to maintain the oxidative homeostasis in such stressful situations, such as intense physical activities that we have just seen, or in the aging process, or in an urban environment which is very rich of stimuli and stressing situation. And to do that, what we need are secondary antioxidants to neutralize free radicals, and we need primary antioxidant to control upstream the production of free radicals. So it's important, we have seen it, that a proper dietary antioxidant supplementation is important, is key to reduce oxidative damage and all the health problems that are associated with oxidative damage. The very last things I want to tell you is that the kind of strategy that we suggest as the optimal antioxidant strategy is to combine primary and secondary antioxidant molecules in order to target several steps in the oxidative pathway and to have a synergistic effect. I thank you very much for your attention, and please feel free to ask you any question or to contact me as you wish. Have a good day.